Hello and welcome to biology class. We are studying topic biochemistry of cell, and in this topic we are studying in details about different biomolecules present in the cell. And so far we reached to the last biomolecule, that is nucleic acids. In last video we have started studying nucleic acids in details. So in this video we will continue with the discussion of nucleic acid. So let us revise what we have studied in the previous video, and then we'll go ahead and understand some more things about nucleic acid. So let us begin. So in the previous video we studied nucleic acid is the polymer, and polymer means the compound which is made up of joining of many monomers. So what are the monomers of nucleic acids? Nucleotides. So nucleotides are the monomers. There is thousands of nucleotides joined together to form nucleic acids. So there are two examples of nucleic acids. Which are those? One is DNA. What is full form of DNA? Deoxyribonucleic acid. Another example of nucleic acid is RNA. Full form of RNA? Ribose nucleic acid. Now we started studying about more details about the monomer of DNA and monomer of RNA. So nucleotides are the monomers. So nucleotides are the compound which are made up of three components indeed, correct? So first component is phosphate group, which is nothing but the phosphoric acid. What more I taught about phosphoric acid or phosphate group? Yes, it is having at center phosphorus and it is having oxygen on all the sides of phosphate. Sometimes here it is having OH group and negative charges on these both of the oxygen. Understanding? Then next component of nucleotide is pentose sugar. Pentose sugar means which sugar? The carbohydrate or the sugar which is made up of 5 carbons, correct? So the type of pentose sugar present in the nucleotide is ribose sugar, and ribose sugar is present in RNA. And which sugar is present in DNA? Deoxyribose sugar. Now tell me, what is the difference between ribose and deoxyribose? D means removed. One oxygen is removed. From which carbon? Carbon number 2. Do you remember numbering of carbon? Very important. Today we are going to use how to number the carbons, or like why we numbered carbons. Then the third component of nucleotide is nitrogenous base. Nitrogenous base, two types, purines and pyrimidines. Purines are single ring or double ring? Simple name. Complex structure means double ring. Okay, 2 rings. And pyrimidine, single ring, 1 ring. I'll just write number 1 ring. Okay. Examples of purines: angel and god are pure. Examples of pyrimidine: cut, pi, CUT. Cytosine, uracil and thymine are the examples of pyrimidine. So today we will study how exactly these components join to form nucleotide, and then how nucleotides join to form nucleic acids. Clear about it? So let us begin. Now let us draw each component of nucleotide separately, and then we will try to understand how exactly joining is done. So the first component of nucleotide is, yes, phosphate group. So this is the phosphate group. Then second component, yes, what is the second component? Pentose sugar. And where is the fifth carbon? Here it is, correct? So this is the pentose sugar. And next component is nitrogenous base. I'll draw pyrimidine ring. So pyrimidine ring we will draw like this, two circles, and purine ring we will indicate just as a single circle. Okay. Now let us I'll just write here nitrogenous base. Now let us see which bonds connect these three components to form a nucleotide. Okay. Now first let us consider about the nitrogenous base and pentose sugar connection, the bond over here. Now when we consider the formation of bond over here in between the pentose sugar and nitrogenous base, they join from the carbon first. See, if you remember, I'll number the carbon again. So numbering of the carbon starts from clockwise direction, the clockwise direction from oxygen. So this is carbon number 1, this is 2, this is 3, this is a 4th carbon, and this one is the 5th carbon, correct? So from the first carbon join to the nitrogen from the nitrogenous base. So nitrogen 4 and nitrogen 9, but in this case nitrogen 9, but we need not to go in that much details. You just need to remember nitrogen joined to the first carbon to form a bond, and this bond is called as glycosidic bond. This bond is called as glycosidic bond. So this is the glycosidic bond. Okay. Now the next connection. So third component and second component got joined to each other. Now it is the phosphate group is to join to the sugar. So phosphate group always join to the carbon number 5, 5th carbon of the sugar. So this bond is formed. Now this is the ester bond, okay, and it is formed with the phosphate group. So it is called as a phosphoester bond. What does it call? Phosphoester bond. Remember the name properly, phosphoester bond. After some time, I am even going to teach you phosphodiester bond, but here it is not di, it is only phosphoester bond. Remember, the bond which join phosphate group of nucleotide to the pentose sugar at carbon number 5. This bond is phosphoester bond. The bond which join this sugar, pentose sugar, with nitrogenous base at carbon number 1. This bond is called as glycosidic bond. Okay. So during the formation of nucleotide, so this is whole nucleotide is constructed. Nucleotide is constructed. So two type of bonds are formed during the formation of nucleotide. See, I'll help you to understand once more. See, phosphate group joined to the carbon number 5 of the pentose sugar, correct? Okay. Now this is the phosphate group, this is the pentose sugar, and this is the 5th carbon, correct? So this joining, which is this bond? Tell me the name of bond. Phosphoester bond, correct? Got it? Then this pentose sugar connect to the nitrogenous base by this bond. What is this bond is called as? Glycosidic bond. Got it clear? So this is the nucleotide. So once again let us write nucleotide and we'll go ahead. Now see, I am writing nucleotide. Nucleotide. Nucleotide is, see, phosphate group, plus second component is pentose sugar, plus third component is nitrogenous base. Got it clear? 3 components of nucleotide. Clear about it? Okay. Now there is another term, one more term is introduced, and this, which is also very important term, which is called as nucleoside. The term called as nucleoside. Suppose I removed, pay your attention properly, this phosphate group and cut from here, okay, the phosphate group I removed, the only two components will remain. Which one will remain? Suppose I removed this, I broke this bond, phosphodiester bond, and removed this phosphate group. Remaining will be only two components. Which are those? Pentose sugar and nitrogenous base. Okay. So when I remove phosphate group from nucleotide, the remaining portion is nothing but the nucleoside. Means nucleoside will contain what? Only pentose sugar, whether it is deoxyribo or ribose sugar, any one of them, and along with that it will contain nitrogenous base, any one of the purine or any one of the pyrimidine. Understanding? So when we remove phosphate group from the nucleotide, the remaining part is called as nucleoside. Nucleoside is also very important while studying this DNA and RNA. They will come ahead. But right now you just need to remember two concepts or two terms. One is nucleoside and one is nucleotide. And even you need to remember the bonds which join these three components to form nucleotide. Clear about it? Very clear? Need to explain again? Very good. Let us go ahead. Now, one more thing, one more statement is given in your book that you need to understand properly. Now, when we consider this nucleotide, there are two terms, chemically there are two terms applicable for the nucleotide. Once, the nucleotide is also called as nucleoside phosphate. Means nucleoside is already there. Suppose I attached a phosphate group to it, it will become nucleotide. That's why one statement is there in your textbook which say nucleotide is nothing but phosphate of nucleosides. Why? How nucleosides will form? Nucleotides will form if I have taken nucleoside and to which I attach phosphate group. Resultant will be nucleotide. Okay, and even they are called as phosphoric esters of nucleoside. They are also called as phosphoric esters of nucleoside. Now this, try to understand it, why they are called as phosphoric esters. Now this thing is nucleoside, pentose sugar and nitrogenous base, to which a phosphate group is attached. Okay, when we attach phosphate group, we attach it with the ester bond, phosphoester bond. So the attachment is by ester bond, phosphoester bond. That's why nucleotides are called as phosphoric esters of nucleosides. So remember, this kind of terms are extremely important when we study for your entrance examination. Clear about nucleosides, nucleotides? Clear about it? Remember it properly because it is extremely important from the entrance point of view. Okay? Clear about it?